It's been over a year since the COVID-19 pandemic began, and now, even with multiple vaccines being rolled out, new strains being popping up that could one day render them ineffective. It is therefore important that we continue to research how the virus enters human cells so that we might find better ways of dealing with it in the future. Entry of SARS-CoV-2, or CoV-2, is dependent upon interaction of the receptor binding domain of CoV-2 spike protein with human angiotensin converting enzyme 2, aka ACE2. It has been established that, when under oxidative stress, these species possess several cysteine residues that form disulfide bridges. Further, when these bridges are reduced, the ability of CoV-2 to bind to ACE2 is completely impaired, suggesting that this method could be useful to combat the disease. However, the effect of individual disulfide bridges is not yet understood, so using molecular dynamics, we investigated what effect the removal of the disulfide bridge between residues C344 and C361 in ACE2 would have on the binding affinity between CoV2 and ACE2. To build the simulations, we began with the PDB file 6MOJ, which contains a 3D structure of CoV2 spike protein bound to ACE2, and then we displayed it using visual molecular dynamics, or VMD. We then used tickle files to manually remove the disulfide bridge between those residues in two different ways, by reducing them to thiols and by mutating them to alanines. We minimized the files using nanoscale molecular dynamics, and ran the simulations using the UWEC Blue Gold Supercomputing Cluster BGSC2. Once we had the dynamics, we analyzed them in two different ways. One was using principal component analysis, which we used to pick out the largest parts of the motion, so it would be a bit easier to observe what was going on. And the other was a measurement of hydrogen bond distance between important residues at the protein interface, operating under the assumption that greater distances would be an indication of less stable binding. For this, we used a simple script to just pull out the average distances between residues, and then we compared those to a control simulation. In both the mutation and reduction systems, PCA revealed the typical hinge motion of the protein complex had a greater range compared to the control, indicating a decrease in stability due to the removal of the disulfide bridge, and suggesting that residues around the hinge are crucial for binding between the proteins. In the analysis of hydrogen bond distance, several residue pairs were found to have an increased average in at least one system, and one pair had a decreased average distance compared to the control. These significant residues are different between the reduction and mutation systems, but most pairs did not display significant distance in any system, and this suggested that the effect of removing the disulfide bridge on binding affinity was pretty minimal. Between these two analyses, we concluded that the removal of the disulfide bridge between residues C344 and C361 in ACE2 had a minimal effect on binding affinity. The inhibition of binding observed in previous studies was likely due to reduction of a different disulfide bridge, or was only observable when multiple bridges are removed. The conserved hinge motion may be an indication that disruption of those residues is necessary to inhibit binding, but further study is needed to determine if reduction of a specific bridge would be able to inhibit binding between CoV2 and ACE2, as well as further illuminate why oxidative stress is so crucial for the interactions between these proteins. Finally, we would like to thank all of our supporters for their aid to us while performing this work. We hope that our small contribution is beneficial to us all. Thank you.